The title of this case study is Whole Brain Imaging to Delineate Spatiotemporal Dynamics During States of Fear and Anxiety in Larval Zebrafish. Fear and anxiety represent distinct psychological and behavioral states. Fear is defined as a rapid reaction to a specific immediate threat, whereas anxiety is marked by sustained anticipation to a general, vague, and future danger. The ability to detect and respond to threats has evolved to allow organisms to avoid harm and ensure survival. And this ability is highly conserved with a high degree of cross-species homology in the structures that regulate fear and anxiety. In humans, fear and anxiety are regulated by a distributed fear network that includes the medial prefrontal cortex, limbic structures, and regions of the brainstem. And the image on the right shows at the top kind of a range of threats and dangers. So the bear on the left is supposed to be sort of a distal threat, going over to the arm, where the shock represents a more proximal threat. And so the bear would kind of correspond to anxiety, something that's more general, you know about it, avoid it. Whereas the on the right, it's an immediate danger. And the important thing is that there's behavioral output depending on whether it's fear and anxiety. There's distinct behavioral differences. So there's avoidance behavior when it's an anxiety sort of generalized threat, going all the way over to fight or flight response when it is a fear or panic. And the image also just shows some structures of the limbic system, the amygdala, the terminalis, and these features are shared in all the mammals and certainly with the other vertebrates. They have homologous structures. So the clinical significance is that this dysfunction in the fear network is implicated in a number of anxiety disorders, such as general anxiety disorder, social anxiety disorder, and panic disorder, which as a whole affect about 13% of people in the US. There are some major research gaps. So the dynamic interplay of the cortical and subcortical structures during fear and anxiety-inducing events is not well understood. The modulatory role of serotonin in the brain-wide regulation of fear and anxiety is unclear. And many studies rely upon immobilized animal models where constrained behavior alters the brain-wide neural dynamics. So in a paper by Lin et al., they examined a number of small animal models that were unconstrained, and yeah, the theory is that there's proprioceptive and vestibular kind of feedback mechanisms during movement and behavior that inform the neural dynamics, and that if an animal is fixed, that it will interrupt it. So the research challenge is obtaining relevant, freely moving animal models that permit in vivo whole brain imaging during states of induced fear and anxiety. So the larval zebrafish provides such a model. So the larval zebrafish have homologous brain structures related to fear and anxiety. And it's transparent and small, so it's ideal for whole brain in vivo imaging. It's a widely used model in the study of a lot of anxiety and psychiatric disorders. For indicating serotonin, a G protein coupled receptor activation based serotonin sensor, which is called GRAB, has demonstrated detection of endogenous serotonin in vertebrate and invertebrate models, and it has a high sensitivity and selectivity for whole brain imaging. It has not been demonstrated in zebrafish yet, but that will be part of the study to validate. And up at in the upper right corner, that is an image of one of the GRAB sensors. So on the left, it's in the inactive state. And then when the serotonin binds to a serotonin receptor, the GRAB indicator has a green fluorescence. And then the third mate tool in this study is differential illumination focal filtering microscopy, which was a technique developed by Kim et al. For whole brain imaging and freely swimming zebra larvae. zebrafish larva. Um, this diff microscopy uses two complementary illumination patterns to obtain an optically sectioned volume, and it is based on the high-low method. And it uses model predictive control algorithm to track and keep animals in the field of view. So this control algorithm has some motion cancellation, but it basically has tracks the brain of the animal, and then keeps it in the microscope field of view, and has a predictive algorithm to sort of based on the trajectory of the animal to keep the whole brain under illumination. The zebrafish is also freely moving. In the image in the right, was from the paper by Kim et al. And you can see the zebrafish is contained in a circular well or pool, and there's a PDMS wall that keeps it contained. So there's one on the bottom and then one on the top that kind of keeps it in a circular, kind of thin cylindrical pool. And this slide shows the structural and functional homologies between the human brain and the zebrafish. So on the left, you can see most of the structural homologies between the human brain and the zebrafish. So in our study, we'll be concerned mainly with what's called the pallium it's the region that is in green. And that is homologous with the human cortex, and specifically the prefrontal cortex. On the right, we see the serotonergic system. So the zebrafish also have a raphe nuclei, and in particular, they have a dorsal raphe nucleus, and it has projections to the pallium that are homologous to the projections of the dorsal raphe nucleus into the prefrontal cortex and the medial prefrontal cortex in humans. So the aim is to characterize serotonergic modulation of brain-wide neural dynamics via the raphe-pallium network, doing induced fear and anxiety in freely swimming larval zebrafish using dual indicator diff microscopy and dimensionality reduction of whole brain activity patterns. The hypothesis is that the raphe-pallium serotonergic network differentiates between fear and anxiety states, which are reflected in distinct brain-wide modulatory spatial-temporal dynamics. So anxiety is marked by high tonic, low phasic activity, where fear is characterized by low tonic, high phasic activity. The research plan. So step number one is to create the transgenic zebrafish line using the crystal transparent mutant. So the crystal mutant was derived off of a prior form called Casper, and it is a mutant variety of zebrafish that is commercially available, that are transparent. So most of the pigment is gone, and the crystal has the retinal pigments are also gone. So it's a very transparent zebrafish line. And this step will involve cloning the GRAB serotonin indicator, and we're using the GRAB G5 HT 3.0, which is a updated version. And because it is green, we want to avoid spectral overlap. So the JR Camp 1A is a red calcium indicator. So they will be both inserted using Toll2 method into embryos 30 minutes post-fertilization. And the plan would be to use 200 embryos for each indicator. The expression of the genetic indicators has to be validated. And once it's validated, the two lines have to be crossed to obtain zebrafish exhibiting dual expression. And so that would be estimated maybe 6 months. The diff microscopy method presented in Kim et al. Also needs to be modified for the dual indication. So in their study, they just had one green fluorescent indicator. In this study, we have to have indicator excitation for both indicators and to avoid the spectral overlap. So one is green and one is red. So there's a 488 nanometer laser for the rad green fluorescent indicator, and then a 561 nanometer laser for the JR Camp 1A, which is the red calcium genetic indicator. The dual band dichroic mirror is to allow both lasers to be sent to the objective, and the dual channel setup. So that includes an emission splitter and filters for the two excitation wavelengths. And in their paper, they used one CMOS camera. A second one would be added here to have a single channel dedicated to each of the colors. So a red and a green one. Oops. The research plan, continuing that, the experimental behavior protocol and paradigm. So the control would be just baseline recording of freely swimming crystal zebrafish larvae 5 to 7 days post-fertilization without introduction of the conspecific alarm substance. So conspecific alarm substance is a chemical a molecular signal that many fish, including zebrafish, exhibit when the epithelium is damaged, and it will induce fear and panic response when they encounter that molecule. So the fear will be induced by introducing the CAS to a perfusion chamber and 10-second pulses. So we have to have a perfusion chamber that runs water through the circular pool the zebrafish is in, so we can cycle out the conspecific alarm substance. The sustained anxiety period will be recorded at the intervals between the fear induction trials. So we'll use a 5-minute period between acute CAS exposure. So that would indicate just sustained, the panic should subside, and the assumption is they're in an anxiety state, just generalized, sustained, preoccupation with generalized danger. Behavioral responses, so those will be recorded and correlated with these states. So the behaviors the zebrafish will exhibit are measured your freezing duration, thigmataxis, so that tends to be clustering, or hugging the side of the wall, the container, and then swim velocity. And these can all be measured with the control algorithm. And finally is acquire whole brain imaging data. So this would be obtaining the baseline data for 2 minutes prior to initial introduction of the CIS, obtain image data for fear anxiety cycle for both indicator colors, and then conduct 10 trials per condition using 20 zebrafish larvae. And then the final stage is to analyze the data. So it would involve using dimensionality reduction techniques such as principal component analysis and detrended fluctuation analysis to reveal the lower dimensional state space of the dynamics. And then you correlate the serotonergic and calcium dynamics to fear and anxiety trials and behaviors, and then identify the brain regions exhibiting activity patterns during states of acute fear and sustained anxiety. So the expected results is that the dual indicator diff microscopy imaging will show distinct serotonin and calcium spatial temporal dynamics using fear states compared to anxiety intervals. So these fear states will exhibit a high amplitude phasic burst-like serotonergic activation of the Raphe-Pallium network, whereas the anxiety intervals will correspond to sustained tonic elevation of serotonin and low frequency brain-wide neural activity. The dimensionality reduction will reveal low dimensional state space models and clusters corresponding to behavioral patterns. So that should be distinct between the fear states and the anxiety states. And the conclusion is that dual indicator diff microscopy and freely swimming zebrafish larvae provides a powerful tool for revealing whole brain spatial temporal dynamics and tonic phasic transitions of the Raphe-Pallium network during corresponding to distinct periods of fear and anxiety. Here's the references, and thank you for your time.